right, so that's my starting chime, so I'm going to get started. So my name is Mike Weiser. I'm a faculty member at Michigan State, and I'm going to be talking to you today about using a taxonomy game to engage students a little more in some of the topics here. So one thing to note as background, experts and novices often see things differently. And one of the studies I think that did a really good job of demonstrating this is show someone a chessboard for about 10 seconds and let them try and figure out if they know where the pieces are when they need to reassemble it. So we have this board here on the left, or alternately, we've got this board on the right. And people who aren't experts at chess find it easier to figure out where all of the pieces are here on the right, just because there are a smaller number of pieces, less to memorize. Chess experts find it easier to reconstruct the board on the left, because the board on the left is a plausible chessboard. The board on the right is very unlikely to ever actually happen during play. So people who are experts and novices are going to view the same information very differently. And that sort of thing also applies in an educational setting. Just because we feel a particular topic is fairly easy or direct to understand doesn't mean that our students, who are often completely new to this work, are going to find the same sort of thing. So with that sort of background, we can start with a nice little pop quiz. Based on this tree on the left, is the frog more closely related to the fish or to the human? And so I'm going to give you a little time to look at this, and we'll talk about that in just a little bit. Now, to answer this sort of question, we're really going through tree thinking. And tree thinking is something that, given that this is the evolution conference, and all of you are experts in evolution, is something that you are almost certainly doing, even if that's not the terminology you're used to. Tree thinking is really an essential skill in evolutionary biology. There's been a lot of research about the ability of novices to get into doing tree thinking. This has been an ongoing debate from a bunch of papers. I'm not expecting you to read these. I just want to demonstrate that there are actually a lot of papers about the importance of tree thinking in education in biology classes, including up into the upper division stuff. I'm not actually going to be talking about upper division students because I don't teach them at the moment. So if we go back to this set of trees that I showed you, we're trying to use this tree over here on the left to figure out if this frog is more closely related to the human or to the fish. So almost certainly what you ended up doing as you were trying to figure this out is trace the most recent common ancestor. So we've got the frog here, we've got the human here. We have to go all the way back here to node X. And that individual is the last common ancestor for the human and the frog. If instead we're trying to trace the most recent common ancestor between this frog and this fish, we have to go further back into the tree. And their most recent common ancestor is individual Y. And because our modern era is up here at the top where we have all of our species, that means that X is more recent than Y. And so the frog is more closely related to the human than to the fish. And so this is the sort of way we will often demonstrate trying to get students to work through trees. And whether or not the students get that is something of an open question. And I think all of us have experienced times before where we thought we had communicated something very clearly in our classroom, and then we give an assessment, and then we see what the students write, and we find out that the information did not make it to the students in quite the form we thought it was going to. So I decided to try and take an approach of seeing if we can use a game to help teach this content. So this is a screenshot of a particular web game called MetaZoo. You can go to it online. Every day it has a new target animal that you're trying to guess. You can also go into practice mode and play as often as you want, and it'll pull a new animal each time it does this. It's got about 400 animals in its database. obviously highly enriched for things that people will actually know about, even if they're not biology experts. You're going to find many fewer beetles in this database than you might otherwise suspect. It's going to be a little enriched for some of the birds, some of the mammals, things that the average public is more likely to know about. So, context of the class that I'm using this in. I teach at a Big Ten university, so this is a Midwestern R1 school, public school. It's considered selective, but it's still roughly 70% of the students who apply get in. It is a giant university, so we've got more than 50,000 students. This is in a 100-level course for biofield majors. So our population and organismal course is the second one in the sequence. So the students have already been through a cell and molecular lecture course. Students have to have had at least one semester of chemistry as well. There are about 200 students per section and 3 to 4 sections per term of this particular class. Each section has one faculty member, one TA, and generally 2 undergrad assistants. And we run these as highly active learning environments. So it'll be a cycle of we get up and talk about something for 10 to 15 minutes, and then we have activities for them to work on in groups while we circulate around and ask them questions. And then we go over the answers to all of those activities, and we repeat multiple times through the course. Sometimes there's some clicker questions thrown in for get some points for participation, but most of the time we're just grading for, are you making a reasonable effort on these things? Our students are mostly second-year undergrads because of the prereqs. We'll also get some third-year undergrads, but it's mostly second-year undergrads. And our evolution is a roughly 4-week module. It comes after the genetics unit. It comes before ecology. I will also tell you of the past several years of this course being taught. I am the sole evolutionary biologist involved in it. Everyone else is either an expert in genetics or ecology. So I have been pushing, if I've only got 4 weeks, to get the general concepts of evolution across to an intro level class. I want them to be able to read and interpret a phylogenetic tree as one of my primary learning objectives. So that brings us to this game, which is a nice gamified tree thinking. So I'm going to show you some screenshots of an example game. So first, you have to guess an animal, and you have no information whatsoever, so any guess is equally valid to any other guess. So on this particular one, I don't know how well those of you in the back can see it, but I guessed mosquito. And it turns out that every time you make a guess, it will show the tree of you are trying to get this target animal. That's why it's in a question mark. Here was a guess I made. They are both members here of Bilateria, so I know that I'm not dealing with another insect. I'm dealing with something actually pretty far from it. So then, as my next guess, I decided to see, well, maybe it's a rattlesnake. And I've determined that now this is amniota. So this has to be an amniote. So that gets me a lot more information. Turns out sometimes you make guesses that don't do anything to advance it. So as an example of that, I guessed a hawk. And so now my hawk and my rattlesnake are both in Soria. So that didn't really teach me much other than that birds and snakes are relatively closely related. I need to go a little further out. This time I guessed tiger, and now I guessed rhinoceros at this point. And so I know it's Laurigetheria. Then I guessed tiger, and I know it's in Panthera, and then eventually I get down to lion. Notice that every time I'm showing you a screen, there's a picture over here of some information about that particular group. There's also a link to the Wikipedia article about that group, so you can go and easily learn more about that particular grouping. Use that to help inform what you want to do next. So I made an extra credit assignment. I don't know about you, but I have found that students are more likely to do something if you list it as extra credit than if you list it as homework. For each one of our modules, we have a homework exercise that is essentially, here's a previous semester's exam. And generally about 2/3 of the students will do that. I made this extra credit assignment, and was worth less than doing the homework, and instead I had about 80% of people do the extra credit assignment. So in the future, I'm debating about renaming all of my homework as extra credit and seeing if I can get better engagement. The extra credit assignment was due a week after the evolution exam. I did a live demo of this game in class on the day where we were introducing phylogenetic trees. And the assignment here, they had to play 3 games, record each guess in order, what they learned from it, and why they made that particular guess. Does not matter if they get to the right answer by the end or not. I just want them to record what they were doing. And then the second part, I gave them screenshots of a few in-progress games. I gave them a list of 5 possible animals, and they had to pick which of them would be the next best thing to guess based on the game they've got. In terms of analyzing this, I used the evolution module exam to predict the final exam score. I added a dummy variable of if they did the extra credit assignment or not. Obviously, the extra credit assignment is not part of either of those 2 exams, so it doesn't directly conflict with it. I had to remove 3 of my 194 students because they didn't show up for the final, and so that wouldn't be a good way to include them in the data. And then 2 things to check. Is this dummy variable statistically significant? And was there a bias in who actually did the assessment? So second question first. The dark red triangles here are the students who didn't do the extra credit. The light blue circles are the ones who did. My x-axis here is their score on the evolution exam. My y-axis here is their score on the final exam. You can see that these averages are 64.3% versus 64.5%. Ran a t-test and my p-value is 0.98. So I'm not particularly concerned that these are different student populations. Looking at the actual linear regression, I have here marginal significance. It's 0.07, but this is 1 class from 1 semester. I'm trying to increase my sample size and see if I can get anything useful. But students who did this extra credit assignment after the evolution exam scored about 2.2% better on the final exam than the students who didn't. And obviously, the final exam is a cumulative one. It's not only dealing with the evolution content, but the evolution content was somewhere around 1/3 of that exam. For the future plans, I have gotten an IRB approval to do this as a formal study. I got the professors in the other sections who have agreed to use this assignment, and I'm going to see what I can find out over the next couple of semesters. And with that, I have time for questions. David.
I didn't know you created it. I've played it. It's kind of fun.
I didn't create it.
Oh, anyway.
Yeah.
But the question I was wondering is, do you think this is because, the effect, which looks real, potentially, is because of the actual content of the thing, or the game, or just because you got students to engage with the evolution material after they finished the evolution module, so they were getting that repetition, that recall effort kind of thing happening?
So I think it's more of the second, but also I will take that in any form I can get it. And I think it's more likely that I can get the students to engage in the material after they've had the exam if it's something a little more fun than just doing another example set of questions, and something that they can get a trivial amount of extra credit for. Yes?
I'm just going to shout from here.
Go ahead.
Do you find, so there's a plant version of this too, which students or people in general have a harder time going through?
I have played Metaflora. I know nothing about, okay, this has a particular inversion on this chromosome. That doesn't help me.
Right. I guess I'm curious about, like, in a course like this, I also teach intro. We don't march through the tree of life. They don't know family names. They don't know group names. Like, do you see a lot or encourage them, or do you see them going and looking up, like, what is an amnio? What is
A lot of them did. I don't march through those group names myself, so that isn't covered in my course. When I was having them fill out the table, a lot of them ended up indicating that they have now learned that horses and deer are not actually that closely related, even though it seems like they might be.
Hello, all. Well, thanks for coming to my talk. I'm Taya. I'm a postdoc at Syracuse University. And today I'm going to be telling you about a study in which I explored student acceptance of common ancestry. So, first off, why be interested in evolution acceptance? Well, as you all probably know, evolution is one of the 5 core concepts of biology, and as such, it's important to any biology-related profession that our students might go into. But nevertheless, studies have shown that a lot of undergrad biology students reject at least some aspects of evolution. And I would argue that in order to make full use of their biology educations, students need to not only understand evolution, but also to accept it. Because if a student doesn't accept evolution, they have little reason to choose to apply it in future professional settings once they no longer have exams hanging over their head. And as citizens, they will be voting on many biology-related socio-political issues, including local and state education standards. So, given the importance of evolution acceptance, evolution education researchers have been seeking to develop instructional resources and teaching strategies that'll help increase students' acceptance of evolution. And survey tools that measure acceptance are essential for this research, because they would allow researchers to explore the underlying causes of rejection and to assess the efficacy of teaching resources and teaching strategies designed to increase acceptance. So, to summarize an entire field of research in one sentence, producing a detailed and accurate measure of evolution acceptance is not an easy task. As evidenced by the fact that researchers have been trying to do this for over 20 years now, by assessing existing surveys and developing new ones based on their critiques of old ones. And nevertheless, a 2022 Research Coordination Network meeting on this very issue concluded there is still much room for improvement. And I have dived into this fray, and I'm trying to build on, what has come before by creating a better measure of evolution acceptance. So, before we do that though, we need to first define what constitutes full acceptance of evolution. So what even is it that we're trying to measure and get at? And secondly, identify the most common ways in which students fall short of full acceptance. So in a recent study, I actually already took care of the first step. So in that study, we generated a widely agreed upon definition of full evolution acceptance by iteratively surveying experts on the measurement of this concept. And we generated this long and detailed definition, which I will not fully read out to you now, but if you want to see it, you can ask me after. But that leaves us to, the second point here, which is what led to my research questions for this study. So, when college students who don't fully accept evolution are asked to evaluate whether it's likely or unlikely that 2 species share a common ancestor, first, at what taxonomic levels do they most commonly draw the line between acceptance and rejection, and does their placement of this line differ between taxa? Secondly, what cues do students most commonly use to decide whether to accept or reject common ancestry? And finally, how does this all look when modern humans are one of the species being considered? So, to do this, I interviewed a bunch of undergrads. So in an interview, we would sit down and I would show them various pairs of species ranging from very closely related to very distantly related. And down here you have a few of the examples that I used. And then, after a very detailed introduction to the study, for each pair, I would ask them, in your opinion, do these 2 share a common ancestor and/or are they related? and why or why not. And at the very end of the interview, after we finished comparing the species, I would ask them, okay, now describe your personal views on evolution and or creationism in your own words. So to recruit students for the study, I first recruited from an intro psychology course by using a pre-survey to find relevant students, and then to increase the religious diversity of my sample, I reached out to various religious student organizations on campus and was like, hey, got anyone who's interested in participating? And after both of these rounds, I got a sample of 9 Christian, 6 Muslim, 1 Jewish, and 5 non-religious students. The non-religious students just kind of slipped in. And after doing the interviews, I did end up removing a couple of the Muslim students from my sample because they actually fully accepted evolution according to the big, long definition that we saw there. And after doing the interviews, I qualitatively analyzed them using a mix of constant comparative method and content analysis. So, before we head into the results, I want you all to take a moment to think to yourself and then maybe chat with a neighbor. So based on your experience, say teaching biology or TAing biology, so of these 3 example species from the interviews, which pairs of species do you think most students, for which pairs do you think most students accept common ancestry and why? And for which pairs do you think most students reject common ancestry and why? And I will give you maybe 30 seconds to think about it, chat with a neighbor, and then just kind of keep that reasoning in mind as we go through the results. Alrighty, in the interest of time, let's wrap it up and dive into the results. Attention, class. Hush. Thank you. Thank you for the participation. On we go. Okay, so for the first research question of basically where do students draw the line between acceptance and rejection, my first finding was that a large majority of students said that species are related when they either happen to share the same genus or the same family. And just so you know, in the interview they were given photos with common names, but I also let them know that they can get additional information about the species if they ask. So generally, same genus or same family, most said, yep, they're related. But then, so moving on to, does this placement of the line differ between taxa? In short, yes. So for most animal pairs, there was a clear switch from a large majority of students saying they're related to a large majority saying they're not related at the level of order and everything beyond. Meanwhile, for the plant comparisons, only a very small majority, like around 60-ish%, rejected evolution or rejected common ancestry at any taxonomic level above the family, but then didn't differentiate between like somewhat closely related plants to super distantly related plants. And then finally, for both fungi and single-celled organism examples, most students did accept common ancestry and were generally unfussed about it. And they were even evenly split on a single-celled eukaryote and a single-celled prokaryote sharing common ancestry, even when explained being like, one has a nucleus, the other does not. So on to the second research question of what cues do students use. So when students said that the pair are related, They mostly relied on shared lay classification and anatomical similarities, especially with regard to body plan. So, for example, the student LP said, they looked at a salmon and a clownfish and said, well, they're both fish, so they have same anatomy, same physiology, it's all the same. They're fish. And secondly, when participants said that the pair are not related, inhabiting an aquatic versus terrestrial environment was one of the most commonly cited reasons where applicable. So, for example, the student Wild the Frog looked at a sea lion and a red fox and said, they're not related because one's primarily based in the ocean and the other one is a land animal. And finally, what about humans? And I've got some particularly interesting findings on this one. So, a large majority of the students said that modern humans and Neanderthals do share common ancestry, and their reasoning often was that Neanderthals are like basically human cavemen, as some of them said. And also, a somewhat smaller majority, but still a majority, said that humans and chimps do share an ancestry, citing our similar anatomy, and mind you, most of these students were religious. And yet, only a small minority accepted common ancestry between humans and non-primates, such as foxes or manatees. And like, this isn't just like a random quirk, because like, for example, one student, Sam, looked at a human and a manatee comparison and said, it definitely makes more sense for God to have put us on Earth as monkeys and us evolved from that than God having put us all on Earth as just fish. So that is a form of special creationism I had never seen before. So, implications for surveys. So first up, many students are willing to accept that closely related, anatomically similar species share a common ancestor, even when they view higher taxa as different created kinds. So a survey should avoid strictly focusing on speciation, as that's not where students draw the line. Second, when considering whether to accept common ancestry between species, students tend to be more attuned to interspecies differences among animals than among other forms of life. So questions should be separate on animals versus plants or fungi. Thirdly, many students find it especially difficult to accept shared ancestry between aquatic species and terrestrial species, regardless of those species' classification. And finally, while some students believe that humans were created separately from all other animals, there's another segment who are willing to accept our shared ancestry with primates, yet reject common ancestry between primates and other major taxa. So we should ask about our common ancestry with both primates and non-primates on surveys. And that is it. Any questions as the next person comes up?
Hi, I'm Abigail Matela from the University of Pittsburgh. So I'm here to talk to you about high school education curriculum to teach students about evolution. And it's called Evolving STEM. So I think we could all agree that there's a need for more innovative teaching practices in evolutionary biology, both because, I think it's important for students to understand evolutionary principles, but also evolution, underlies everything, our understanding of the natural world, and also things that are important for humans in particular. So I think most students are still learning about evolution with Finch adaptations to food sources, which I think is really interesting, but they can just read about that, right? So we think if you look at microbes, so bacteria, because they reproduce so quickly, we can actually watch evolution and selection happen in real time. So our goal is to bring this to students with an authentic research experience, because this is a really powerful way to engage students in science. These kind of experiences both improve learning and build relevant lab skills. We're hoping that they're sparking more interest in STEM careers, improving STEM diversity, and I think on top of that, what I'd like to share is that I feel like student research can actually feed back to contribute to our own research in the lab. We designed our program based on our lab research looking at bacterial biofilms. So if you don't know, most bacteria live collectively as a biofilm, or just a community of bacteria that are together, in this case I'm saying on a surface, and produce a matrix to protect them. So there are different niches in the biofilm. So if you select for bacteria to improve biofilm formation, there are a lot of different areas where they can adapt, and the populations will diversify. Biofilms are also important because they're a ubiquitous part of every habitat on Earth. So they help support plant growth. They're an important part of aquatic ecosystems, even us in our human microbiome. But they can also have negative impacts, like coating pipes and contributing to both corrosion and dispersing pathogenic bacteria into our water systems. The plaque on our teeth, that is a biofilm. And probably most importantly, in disease, so in this case, thinking about diabetic foot ulcers, and if they have bacteria that get infections and create a biofilm, you can have these chronic wounds that are really hard to treat. So how do we do this in the classroom? So we give the students bacteria, safe bacteria, Pseudomonas fluorescens, and they have these little plastic beads, so that's the surface for the bacteria to attach to and grow on. So students will put 2 different colored beads into a test tube with media, add bacteria, and then allow the bacteria to grow overnight. Bacteria will reproduce. Some of them will attach to that bead and make a biofilm. And then the next day, the students will use an oppositely marked bead, fresh bead, new media, and they'll transfer the biofilm covered bead to that new test tube. And they do this really for just 4 days, in as short as 4 transfers. And as those bacteria are reproducing in the test tubes, and they have to both move off an old bead, attach to a new bead, and make a new biofilm. If you have mutants that are better at attaching to the bead and making a biofilm, they should increase in frequency in the population over time. So in this diagram, we're just looking at the black bacteria are the ancestor, and new mutants are in color, so the yellow one better attaching to the bead, and it increases in frequency in these populations. So students can actually observe evolution happen in 2 ways. So one is at the beginning of the week. If they look at their test tubes, you won't see very much happening on the side of the test tube walls. But by the end of the week, typically, these bead-selected populations will have a layer of biofilm, which is easily observed. The other way, and the kind of more fun way, I think, is that they'll plate the bacteria 2 times during the week. So in the beginning, all the bacteria will make these round, smooth, we call them smooth colonies. But by the end of the week, they usually see a diversity of phenotypes. Most often these wrinkly phenotypes, but also there'll still probably be smooth colonies in these populations, and then there's some other different colony morphologies that we can see. So what are the mutations that we know of that underlie these adaptations? So most often we're seeing these wrinkled colony morphologies, and they're typically mutations in this WISP pathway. And interestingly, we can actually relate that to a pathogenic bacteria, Pseudomonas aeruginosa, and how they adapt in chronic wounds because they make they'll collect similar colony morphologies, and the mutations also occur in the same pathways that we see in the safe classroom bacteria. So student discoveries both support things that we already know about how bacteria adapt in wounds and in making biofilms, But they also, we've had newly discovered pathways in some other genes that we haven't seen before, so I think that's really interesting. And then we also do have a bioinformatics module that teachers and students can use if they want to explore what those mutations are. So also, I'm going to show you a little bit about how the WISP pathway works, and we do run a summer teacher training program, and one of the teachers actually in the summer program made this animation, and I think it's really good. So how does WISP work? So an environmental signal will result in conformational change to WISP-A and allows it to be methylated by WISP-C. So WISP-C transfers the methyl group to WISP-A. This signals WISP-E to catalyze the phosphorylation of WISP-R. WISP-R is a diguanylate cyclase that catalyzes the reaction between two GTPs to produce cyclic diGMP. And cyclic diGMP is a secondary messenger molecule that signals for increased biofilm formation. WISP-E also phosphorylates WISP-F, which can then remove the methyl group from WISP-A and then shut down this pathway. So this is how the pathway would normally work, and one of the more common mutations we see is in WISP-F. So again, you're getting methylated WISP-A, phosphorylated WISP-R. This is producing cyclic diGMP, making more biofilm. If you have a mutation to WISP-F, in which it is no longer able to remove this methyl group, then you're just keeping the pathway in a constantly on state, making lots of cyclic diGMP, lots of biofilm, and in this case, it makes a lot of cellulose, and so you get this wrinkled colony phenotype. So our program has been adopted all over the country, but mostly so both in high schools, now in middle schools in Pittsburgh, and at the college level. We've mostly focused our research on Pittsburgh public schools, so local schools in our near the university. So what we wanted to find out, one thing we're researching is if students perform these experiments, do they have improved interest, self-concept, self-efficacy, and confidence in science? And so one way we do this is to give students pre-surveys before they have done the experiment and then post-surveys after. We're asking them these questions. So things like, I can become good at science. And then there's a 4-point Likert scale, and so students will answer that based on their Likert score. And then we're averaging the pre-surveys and then take averages of the post-surveys and seeing if there's a change. All this data I'm showing you comes from 114 students, about half girls and half boys. They're taking introductory, so like 9th grade biology. And we have a pretty diverse student population, so I'm sure you can't read. The pie chart is racial and ethnic data, and I'm sure you can't read the names, but orange is white students, and all of the other parts of the pie are underrepresented students in science. So a lot of data, but I'll just point out a few things that I think we're seeing overall fairly good improvement in how students see themselves as scientists after they get to do a hands-on research experiment. The statement, I am a scientist in particular, we see a significant gain in how students see themselves as scientists before doing this experiment and after. So I think we're pretty excited about that. So if students are doing science, they see themselves as scientists. the statement I often ask questions about science, also seeing improvement there. That's significant. And I like learning about science. So that overall student population, we're seeing improvement there. Oh, and so to direct you, but the dumbbell plots, the colored ones are their pre-survey averages. The black are post-survey. Gray is for all students. Purple is for girls, and orange is for boys. So one thing I wanted to point to is that actually there are some questions where we saw boys, their pre-surveys, they actually go down after doing this experiment. So they thought they knew more than their friends about science beforehand, but afterwards, not so much. So similarly, we asked them about lab work, which they like more to start with anyway. And again, we're seeing improvement in things I often ask questions about science labs. And then, actually, again, in a similar question, I know more than my friends about science labs. We do see a decrease in how boys think about that. But overall, mostly, I mean, these aren't, mostly not significant changes. But again, hopefully we'll have more data over the years. And as we have data from more students, maybe some of these will be significant. So I just want to finish by saying, I hope what I've shown you is that authentic research enhances student identity as scientists and hopefully can promote STEM pipeline diversification. And that student experiments actually directly contribute to research. And thanks to the lab. And I will point out especially undergrads who have been helping me, Colton, who is here, and he's giving a talk this afternoon in Experimental Evolution 6, and Sachin and Sarah. So thank you.
Hi everyone, I'm Ellie. I'm a PhD student at the University of Bath, and today I'm going to be talking to you about fruit fly citizen science, better engagement when the data collection matters. I am sorry if you've come here for fruit flies, I don't actually talk about that much in this. So my PhD ultimately is looking at the relationship between citizen science and science capital. So for those of you who aren't too familiar with citizen science or science capital is basically a metric of how all your experiences, attitudes, values about science all come together. And it is measurable. So it's quite linked to social capital, if you're familiar with that. So lots of experiences feed into your science capital. So it's things you do in school, science experiences you have at school, science experiences you have outside of school, so like museums, going to zoos, that sort of thing. It also is a lot to do with your background, so if your parents are well educated, may have encouraged you to take part in science. So lots of things that aren't quite as measurable from sort of like a survey point of view. So why I'm interested in citizen science, it's because so basically, citizen science is getting the public to participate in research in some capacity. Often this is used quite indirectly. So if you're familiar with like bird watching apps, that kind of thing, where people who are already hobbyists, already involved, they will upload their data. And this is used in a surprising number of publications. But it's still quite indirect. And it's very much sort of like getting people who are already interested in science to help contribute to science. So I felt like there was potential here for it to be used in a lab setting and to get more young people involved in what research is actually like. Because they have done surveys of the typical participants of citizen science, and they do tend to be highly educated, older, white individuals with lots of free time and already established interest in science. And I just think there could be an untapped market here to help people with lower science capital sort of get more of a research experience. So there's lots of reported benefits of participating in citizen science. And I'm sort of thinking if we more thoroughly research how participants of citizen science view their own experiences. A lot of citizen science research has been on how we as researchers benefit from citizen science and why we might want to do it, but not that much has been on why people would take part in citizen science. So I'm sort of hoping that my research could go into designing more effective citizen science that serves more purpose than collecting data on large scales, as useful as that is, but can we develop it in a way that is as beneficial to the people taking part as it is to us? So I think that might be quite small. In a roundabout way, what my project aims to do is understand how citizen science activities contribute to science capital. This has sort of been touched on, but not to a great extent. And then through that, I want to demonstrate alternative ways of evaluating engagement in addition to learning. Lots of people will do, especially with young people, interventions that are focused on like knowledge gain, and also obviously very important, but as you'll see in a minute, lots of people get more than just learning from participating in science. And then ultimately, it would be great if this could feed into increasing accessibility to higher science capital and greater diversity in STEM careers. So what did I actually do? So I have developed a workshop that I take to Science Oxford, which is like a science outreach centre, and they're running 15 to 17-year-olds who are already interested in a STEM career. So they're already there for a STEM careers week. But as far as I'm aware, I'm the only researcher that goes. Basically, I take my fruit fly lab experiments that I'm developing. So to touch on that very briefly, I run nutrition studies looking at self-medication. So I run about an hour-long workshop with them, introducing them to the experiment. They do take part in the experiment, but importantly, I really wanted to highlight what the process of research is like, so I wanted to get them involved at more processes. So we actually look at the data in real time, which is quite interesting, and I sort of get them to think about like, what was good about this experiment, what would you improve? So yeah, we do that, and then I basically take 8 of them off for a focus group with me afterwards, where I get them to do a few activities around science capital and citizen science. So I'm mostly focusing on the citizen science part of this. So on the screen, this is not an exhaustive list. These are reported benefits of partaking in citizen science from a whole range of literature on this subject. So for example, people report that they like working with others, that they feel personally empowered, they like the opportunity to network with other like-minded people or role models, and some of them just say that they find it fun. So it's not all sort of like career-based or anything like that. So what I get them to do in this focus group is, as a group, I get them all to discuss, since they have just taken part in citizen science, I get them to discuss which of these benefits, there are more, I just couldn't fit them all on the screen, which of these benefits are most important to them. So it does create some interesting discussions, because obviously these are all individuals with their own science capital and all have slightly different reasons for why they would take part in this. So I would just like to caveat all of this and say this is very early provisional data, but I thought I would just give you some quotes from this focus group. So first of all, what did they value the highest? So it wasn't actually what I expected. They actually valued having fun and collecting impactful data. This came up consistently. So if you can't read that, it says, I'd say having an impact would be the most important thing, because that's like the whole purpose. And if you know it's going to be useful and you know it's going to have an impact, then it makes people want to do it. So I wasn't quite expecting this to be something that, it just kept coming up time and time again, that actually once they understood that the data they were collecting was for my PhD, they seemed to then realize, like, oh, actually this is really important, this would make me want to do more of it. What I'm doing is not just a trivial thing, it's for an actual purpose. And having fun, they did rank this. I think that was actually number 1. It's important to me to pick what I'm going to do in the future, so I'm considering what I enjoy now into what I want to do later. So I guess you can also highlight that it was still very STEM careers based, just based on the nature of the week, and I will discuss that in a second. So a lot of this was still in the context of they were thinking about their future career. So what's less valued? This was quite interesting. So they didn't find that working with others was necessarily a benefit, because you can get the opportunity in loads of jobs, is what they were saying. And they didn't think that understanding the experiment process would really add anything to their experience, which I also thought was interesting because that's what I wanted to give them through my workshop. So I was maybe need to go back and rethink that. The one I think that stung the most was, I don't think communicating with research has any impact on your reasoning to want to participate in a study. That was quite difficult to remain impartial when they were talking about that one. But it's still interesting because I think it's something that people cite as a big draw to partake in these kind of things. Like, oh, look, you get to communicate with researchers directly, and these are young people who might want to go into science. But actually, well, they didn't think communicating with me had any impact on their reasoning to want to participate. But maybe it's me, I don't know. And they didn't think that a sense of independence was that important. They think you can get independence in anything you do. So I think a lot of the sort of more, I guess, like general sorts of skills you would get, they felt they could get from lots of experiences that wasn't necessarily unique to citizen science. So it was quite helpful to understand that. But like I said, this is very provisional. This is from one focus group. So what I plan to do next is, obviously I plan to repeat this. I'm going back next week, I think, to do another one. But also, so like I said earlier, this is a group who are already interested in a STEM career. They are already STEM-oriented people. So arguably they are a group who is likely to have higher science capital, or it's not necessarily like the group that, in an accessibility sense, I'm trying to reach. So I was really struggling to think of groups where they wouldn't necessarily have this science focus, that I would then be able to recruit to do like an hour-long workshop with me and then an hour-long focus group. But actually someone pointed out that going to like the Girl Guides group, so like Scouts, that kind of similar group, they're not necessarily STEM oriented. So I am doing some workshops with them at the end of the month, which I think will be quite a helpful comparison, see if the same sorts of things come up or whether it is different priorities. And I did also get all my focus group participants to carry out a science capital survey, which I have not evaluated yet, but that is still important to understand what the science capital of that group was. So yeah, lots of future work, but I just thought it was an interesting start, so wanted to share that with you today. But yeah, thank you very much for your attention. I'd like to thank the Priest Lab and my funders as well as Science Oxford, and I'm happy to take any questions. Yeah?
Yeah, given that you did focus groups, do you think that group dynamics might have really changed the answers? About one or two people giving an answer and then everybody else being like, yeah?
Yeah.
Or like, did you observe anything like that?
Yeah, so the question was about group dynamics in a focus group, where there are people who, people who were answering more questions than others, and everyone else just agreeing. That was actually really challenging because I'm not a social scientist, and so like this was my first time running a focus group, and I found it interesting. There are always those dominant characters, and I did find that, and so I was having to sort of remember, like, as the person conducting it, because I was transcribing it, to say like, you're all agreeing, or you're nodding, so that I had sort of an idea of who was actually agreeing but not saying anything. But yeah, there were definitely people who, I think there was one person in that group who spoke once. So I think it's challenging to do a focus group. I think in hindsight, I think I probably would have done interviews if I were to do it again, just to avoid that interaction. But it's quite interesting to look at the interaction between people as well. and how their opinions are influenced by other people. It is interesting, but it's complicated. Yeah?
Great talk. I was wondering if you had any way or plans to follow up on retention. Like, was there any way to track what, if they were to continue in a citizen science program or continue in a STEM career? I know it's kind of hard with minors and students, but if
Yeah.
there's something that you can do to track
Yeah.
for peace.
So the question was about retention. So would I be able to track then what they went on to do? I don't have that sort of capability. I think I would have to go through a whole another round of ethical approval to do that. I would love to, but at the same time, my supervisor, Helen Featherstone, she said something to me ages ago that has always stuck with me, in that one intervention isn't necessarily going to change the career trajectory of someone's life. And I think it's quite important to remember, like, it's helped me a lot, take a lot of the pressure off of designing what I do. But I think it would be interesting to see, because also it's difficult to get retention in longer term citizen science. I think that would be more worth tracking. I don't think tracking a one-off thing would work. But the people who consistently participate, it's hard enough to get people to consistently participate in citizen science. So tracking that retention, I think, would probably be of more value. But I would really like to. It would be interesting to see where these people go. They already had an idea of what they wanted to do, and it was very varied. Some of them were like convinced they were going to be a doctor, and others didn't. So it would be interesting to see where they went. But it's a whole ethical nightmare, especially with people under 18. Cool, thank you.